function of neurons are characterized by their ability to make connections. So the more complex they are, meaning the more intersections they have, the more branchings they have, the better they'll be functioning. So reduction in this area would actually mean they're functioning less. So taking a look at the basal region of the pyramidal neuron, the overall basal length, number of intersections, and the number of branchings are statistically the same. So there's change occurring in the apical region, but no change occurring in the basal region. And this is rather peculiar. So the overall conclusion is that there's shrinking in the apical region, but no changes in the basal region. So maybe it's just a case where shrinking or reorganizations of the dendrites only occur a certain distance from the cell body, which is still an open field for argument. With that change in apical and apical length, the researchers were curious as to whether or not there would be changes in the number of dendritic spines present. They noticed that the apical spine density and the basal spine density for both groups were statistically the same. However, with the shrinking that occurred, the apical dendrites getting shorter, it was expected that there would be fewer apical spines present. So that's why you have less apical spines, but it doesn't mean that the cells are producing less spines, just means that they got cut off because the apices got shorter. And again, supporting this idea, no changes in the number of basal spines would suggest that there was no shrinking occurring there. So let's take this puppy home. If we take a recap to our initial question, how will circadian disruption impact normal physiological and behavioral functions in mice? Then let's take a look at what we've learned. Chronic circadian disruption has led to increased body mass, which is either the cause or the effect of increase in leptin and insulin concentration. Previous literature has supported the notion that insulin and leptin are responsible for the remodeling of neuronal structures, and this could also be a direct effect of stress from the circadian disruption. The remodeling of the neocortical neuronal structure leads to a decrease in complexity of the pyramidal neurons. And this overall decrease leads to a decreased or impaired ability to perform higher order functions. How exactly this translates into human subjects is not quite clear. But the hope is with models like these, researchers will be able to see potential impacts of chronic circadian disruption on workers who are placed in these situations. Healthcare professionals and pilots and these type of people, and the risks that are posed to them because of their shift working.